Hello, everyone. Thank you all for joining us for our April Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Hereditary Disease Foundation. To start, I want to thank our sponsors for this webinar, Ionis Pharmaceuticals and Unicure. Also, a big thanks to everyone who supports the HDF. Truly, it is because of you that we are able to fund exciting science that is transforming what we know about Huntington's disease. And of course, a big thanks to all of you for being here. Today's webinar is about genetic modifiers. These are small changes in someone's DNA that can speed up or slow down the timeline of when someone with a gene for HD will start to develop symptoms. And some of these genetic modifiers can have really big swings, changing when someone will get symptoms by perhaps up to 10 years or more. So, learning about them and figuring out if we can control genetic modifiers can be a real game changer for developing treatments against Huntington's disease. To learn more about genetic modifiers, we are going to be hearing from Dr. Gabby Baumus and Dr. Zach McLean. Dr. Baumus is having some technical difficulties, but hopefully he is able to join us soon. Dr. McLean is an instructor of investigation at the Center for Genomic Medicine at Mass General Hospital, Harvard Medical School, and the Broad Institute. He works in the lab of Professor James Gusella, who is a pioneer in HD research and is actually going to be our May webinar speaker. Dr. McLean's research focuses on in vitro modeling for genetic modifiers of Huntington's disease. Thank you so much for joining us to share your work. Before I pass it over to Zach, I want to remind everyone that you can ask questions in the Q&A box throughout the talk. So type them in so that you don't forget them. We should have about 20 minutes at the end for live Q&A, and we will address as many of those as we can. Also, a reminder that this will be recorded and available on both our website and our YouTube channel in about 2 weeks. So if you want to watch it again or if you want to share it with someone, it will be available for that. Thank you all for being here, and I will now pass it over to Zach.
Thanks, Sarah. Okay, so today I'm going to talk about targeting PMS1, one of these genetic modifiers Sarah was mentioning, to stop somatic repeat expansion in Huntington's disease. So first, who am I and where have I come from? So I'm originally from this place called Tauranga in New Zealand. This is where I grew up. And my first introduction to science was probably through my parents when my father had some little experiments breeding flowers, the horticulturalist. And then my uncle actually gave me a tour of where he worked, which is this government research institute, Plant and Food Research in New Zealand, where I met many scientists actually, and that really inspired me meeting these scientists working on plant breeding. So I went on to study at the University of Waikato in Hamilton. Then eventually I went on to do a PhD at the University of Auckland and in Hamilton at the Ag Research, which is another New Zealand Government Research Institute. There I worked on reproductive technologies for livestock breeding purposes. Basically, I was doing genome editing. I was very excited about genome editing technologies, which is an emerging technology for modifying DNA. And so I was doing this and then using somatic cell nuclear transfer cloning, like Dolly the sheep, to generate genome edited sheep. And you can see on the right there, it was one of me feeding one of these cloned sheep. However, I was also very much, I wanted to work on helping people as much as possible. And so I knew that for my next steps of my career, I wanted to work on biomedical research. And one of my supervisors for my PhD, Russell Snell, has been also a really long time HD researcher, has done very important work helping to figure out the connection of the repeat expansion in HD. And so I was familiar with HD through him. And then for my postdoc, like Sarah said, I was very fortunate to have an opportunity to work with Jim Gusella at Massachusetts General Hospital in Boston. And so this has been a place that is really done through Jim and Marcy McDonald and other collaborators at MGH and other HD scientists across the world as well. Done very important work making the connection, figuring out the gene that causes HD, and then as we'll talk about today, genetic modifiers of HD, which is hopefully going to lead us to developing effective therapeutics for Huntington's disease. So now I start off as a postdoc, which is the position you get after a scientist, and now I'm an instructor at MGH and Harvard Medical School. So early on in my postdoc, I actually got a fellowship from the HDF to work on HD, which was really great and helpful for my career, and present some of the work from that today. So what's our philosophy overall in Jim's lab? What do we at Jim's lab and the Center for Genomic Medicine? So we overall work with a philosophy of the genomic medicine cycle, which is we start off with patients and families understanding the genetics that cause the disease and in our case modify Huntington's disease. Then understanding those mechanisms, how they are altering those different signs of the disease, and then how we can figure out how to use that information to actually transfer back to patients using those mechanisms to try to develop therapies and therapeutic strategies so that we can find treatments for HD. So we know HD is caused by a CAG repeat in the Huntington gene. And we've long understood that the length of the CAG repeat, how many CAG repeats you inherit, is correlated inversely with the age at motor onset. So the longer repeat you have, the earlier the onset of symptoms. However, within a single CAG repeat inherited length, there's a huge variation that spans decades, as you can see on the right here, for when that onset of those symptoms will begin. So a really cool, I think, experiment that was done, led by the GEMHD consortium, was to say, okay, what can we figure out what is the causes of this variation by figuring out, is there associations with the other genes in the genome that might help explain why there's such dramatic variation in onset. And so that's what this plot shows. You don't have to necessarily understand this other than say that each of these different colors is one of your chromosomes in the DNA. And this is called a Manhattan plot, where there's these skyscrapers that indicate a signal that has emerged of another gene that is associated with a phenotype. And in this case, we can look at a couple of different phenotypes associated, so these are, for example, the age of onset of the disease. Then we can say, okay, what variations in the genome are associated with this phenotype? And this, I think, was a really amazing finding where there was many genes that have now been associated across 12,000 or so people with HD. And these have converged, I'll say majority of these have converged on really a single pathway, which is kind of rare in human genetics, I think, that things have coalesced relatively to a mechanism that points to DNA repair in the cell. So the cell is constantly repairing itself. There's always mutations that are occurring in your DNA, and these mutations are being repaired. And so many of these genes, PMS1, MLH1, MSH3, PMS2, FAN1, LIG1, OD1, are all associated with the repair of the DNA, and specifically on one pathway of DNA repair, which is called mismatch repair. So PMS1, MLH1, and MSH3, PMS2, these ones are all in mismatch repair when you have, for example, a little the insertion of a wrong nucleotide, and then there is a mismatch. So this needs to be repaired in the cell. So overall, we can say we've arrived at a mechanism of disease, and we know from various studies that these this is associated with a process called somatic repeat expansion, which I'll explain. But a couple of these modifier genes have really emerged as excellent therapeutic targets for slowing down HD, because the variations in these genes, small changes over the lifetime of the patient, have resulted in this delaying or hastening of symptom onset. That means that if we can target these genes, therefore we can hopefully recapitulate that and, for example, delay onset by lowering these genes. So MSH3, PMS1 are both excellent therapeutic targets, because we know from various studies that lowering these genes is associated with delayed HD onset. Yeah, so what is this concept of somatic repeat expansion? So we know that in Huntington's disease, you inherit over 35 repeats causes the disease. So here's an example of these 40 repeats. And this, if we zoom in on a piece of the brain and then look at different neurons in the brain, different cells of the brain. So what happens is there's a mosaic situation where different cells have different CAG repeat lengths, and some cells have, over time, accumulate very long repeats. And so you get the situation where there's some cells with short repeats and some cells with very long repeats that expands after birth over many decades. And so a recent study has really nicely demonstrated this, where this is a picture of 6 different brain donors that from people with HD, showing different cell types here in the different colors. And you can see that on the x-axis here is the length of the repeat, and the CAG repeat expands in the cells that are the ones that are vulnerable and are lost in HD, these SPN cells, whereas the other cells that aren't lost do not have this somatic expansion of the repeat. This paper also showed that when you zoom in at the cells and look at what's the signature of these cells. So overall, there is a signature that is very intriguing, where it doesn't look like there is major differences in the cell signature, disease signature, until the repeat expands to approximately 150 repeats. Then there is a signature of the disease that continues to increase as the CAG repeat length in the cell gets longer and longer, up to like 1,000 repeats. Somewhere in this range of 500 or so, ultimately that is a toxic threshold in the cell is lost. So why this? So overall, this leads to a understanding of HD where it has 2 sequential components. First, the somatic expansion of the CAG repeat, as I described before, that happens slowly and asynchronously across different cells. So then if we zoom in on a single cell here, so let's think about this one cell over time. So if we think about this cell over many decades, and you can see that the CAG repeat length, which is indicated in the red line, slowly expands and then begins to expand more rapidly. Then the idea is once it reaches a certain CAG repeat length threshold, a toxicity process is triggered, and that ultimately leads to the loss of that cell. So why that is super interesting is this really points towards stopping the somatic expansion of the CAG repeat as a key therapeutic idea. So how would this work? How would a therapeutic that targets somatic expansion actually help in Huntington's disease? So, for example, if you were to start taking a therapeutic that delayed the rate of this red line, overall, you can see in this dotted line indicated here, this would slow the rate of this growth of the cell and then slow down the time until it takes to get to this toxicity threshold. And during this time, the hypothesis is that this is not actually toxic, and so the cell is fine until it reaches that threshold. Therefore, this prevents losing the cell. Okay, so now let's talk a little bit about therapeutics. So first, let's look at another successful therapeutic in another disease, spinal muscular atrophy. So now, of a decade or even longer ago, various companies developed small molecule splice modulating drugs. So these are drugs that are designed against little bits of DNA, basically, and can disrupt the message that is produced from a gene and prevent it turning into a protein. So basically wipes out the that gene is the way you can think about it. So these are excellent medications because they're oral medications that you can take. They have broad distribution, including deep into the brain across different regions, which is important for Huntington's disease to where the whole brain is basically affected. There's certain regions that are, as we know, that are very affected, but it's great to get a broad distribution. And so for spinal muscular atrophy, Novartis and PTC Therapeutics had developed branaplam and risdiplam, respectively. So these ones, so risdiplam is now an approved therapeutic and has had really impressive results for people with SMA, and they respond well after this treatment. So it's been a great, successful therapeutic. What's very interesting for HD is that branaplam many years ago was shown by Novartis that it also lowers Huntington. So it's one of the more sensitively hit targets of branaplam. So then they started a clinical trial for Huntington lowering. Eventually, as I'll go on, I'll explain later as well, this trial was stopped. But during that time, we actually decided to look into branaplam. We were interested in how different patients might respond to this, but we also discovered along the way, amazingly, that branaplam and risdiplam actually also target PMS1, which is that super important genetic modifier, which is also one of the key potential therapeutic targets that we know that if you lower this, that would be a great thing for delaying HD onset. So when I figured this out, I was amazed, honestly, when I saw this gene in the list of genes that was hit by branaplam. So we showed that that these drugs, branaplam and risdiplam, they result in disruption of this PMS1 message, and this occurs at a dose-dependent way where more of the drug results in more of this, what's called a pseudo-exon, poison exon being included in this message. This results in lowering of the PMS1 protein, which then you then lose the function of that gene product. So we then showed that PMS1 so we then we made cells that are a model for somatic repeat expansion, where we can look at this process in vitro. That allows us to add the drug in vitro, to see if it would have the desired effect, which is to stop the repeat expanding over time. So we showed that when we treated the cells with branaplam, it actually lowered the rate in which the repeat expanded in vitro. We also showed that this was due specifically to PMS1, making the direct connection for PMS1. So as I mentioned, branaplam, the study for branaplam was halted due to some negative side effects that people taking the drug, people with HD that were taking the drug, experienced some peripheral neuropathy. So Novartis ended the clinical trial that was for this Huntington lowering. But overall, PMS1 targeting then becomes a great target that you can design a therapeutic for, a new therapeutic. And so what's going on with the splice modulating compounds and the different companies? So this is now really an active area of therapeutic study. There's lots of companies working on this. And so this is some of the most advanced companies with these small molecule drugs these ones are for Huntington lowering. So PTC Therapeutics, now in partnership with Novartis, have this drug Votoplam, which is for Huntington lowering, which targets this second phase of the disease. The toxicity component is the idea. And they're testing to see if lowering Huntington in this way will be beneficial for HD symptoms. This is an ongoing clinical trial due to end in 2027. A new company, Skyhawk Therapeutics, in this area is about to start a new clinical trial, also for Huntington lowering, that is due to start next month. So I think very excitingly, there's also companies that are working on this PMS1 lowering as a target as well. These are great compounds to design against targets because you can really, you've got like a great thing to optimize on. So one company, Argenta, is developing these compounds very specifically just to target PMS1 without targeting Huntington, with the idea that stopping the repeat expanding and preventing the toxicity in the first place is a way to delay HD symptoms. So this one is very specific to PMS1, and they've shown that their drug has in vitro and in vivo target engagement. So I guess watch this space. Hopefully, I have no idea how far away they are from a clinical trial, but the signs are positive. Another company, Revere Therapeutics, is going with a strategy of developing a compound that targets both Huntington and PMS1 simultaneously to try to target both phases of the disease. They're aiming, I think, I was speaking with them recently at a conference, for more PMS1 lowering than Huntington lowering. Yeah, so I think they've identified some compounds and working on it. So overall, I think this is very exciting news, this area of somatic repeat expansion, because it links together potentially not only Huntington's disease, but other repeat expansion diseases with a shared potential underpinning of disease pathogenesis. And why that's great for people with this disorders and HD is that if they have a shared mechanism, that creates a much larger and great therapeutic target. And so more companies are interested because they can potentially make a drug that works for a whole bunch of other repeat expansion diseases. So I think now there is quite a lot of therapeutic interest. Honestly, more companies working on somatic repeat expansion than I can keep track of. So hopefully we'll have some great progress soon. Overall, acknowledging all my colleagues that were co-authors on the publication I talked about, and the funders that funded us for this work.
Thanks so much, Zach. That was awesome. We have quite a few questions lined up, so keep putting your questions in as we continue this conversation. Unfortunately, it looks like Gabby's still not here, so we're gonna move forward with the Q&A with Zach. So I think I'll just start at the top here. Could it be that somatic expansion is not the trigger of onset, but a biomarker for it? Since onset can happen before somatic expansion, is there any animal data to show that stopping somatic expansion can prevent symptoms?
So I would disagree with that slightly. I think that there's not enough time to go through all of the evidence, but there's various genetic evidence that points towards this Huntington's disease being a two-step process where you've got the somatic repeat expansion and then a threshold event. So I would disagree and say that I think it's a first step in the disease. But definitely it's like either way, if you're in the other mindset, still in that case, I think stopping somatic repeat expansion is going to be an important component.
And they also asked if there's animal data. So maybe you could talk a little bit about
Oh
Vanessa's
yeah, yeah,
work
yeah.
and Jill's work.
Oh yeah. I forgot about that. So there's definitely lots of data showing that when you stop somatic repeat expansion, one, there's like a signature that is seen on the what's called the transcript level, which is like the expression of all of the genes in the genome. So there's recent work, for example, from Jill Bates and William Yang's lab, um, and some of my colleagues, Vanessa and Ricardo Moro Pinto. So that when you stop the somatic expansion of the repeat, it prevents this disease signature in animal models. There's also evidence that, for example, what happens when the repeat expands even longer is that you get aggregations forming of the Huntington protein, and that stopping the somatic repeat expansion prevents these aggregations forming. So there's definitely evidence that preventing somatic repeat expansion is improving the phenotypes of the disease.
This is a kind of a similar question they asked. Could targeting these genes revert somatic expansion? So maybe they're talking about contractions. Could you talk a bit about that?
Yeah. So we know that the repeat is expanding over time, but one interesting thing is that there has been some observations that the repeat can contract, so it can get smaller. So we're still now trying to understand the genetic underpinnings of this process, but we know that this can be driven by, for example, a CRISPR-Cas9 nicking at the repeat can lead the repeat to contract. So you could imagine a therapeutic strategy where even though the repeat has somatically expanded to this really big level, maybe we could figure out a way to send it backwards to a shorter length. Is another idea, not just stopping the repeat expanding, but going backwards. So that's kind of like I guess, a long-term aim for all of us to work on.
That would be super cool. And Christopher Pearson is someone working on that with small molecules.
Yeah.
So here's a question that's asking about overexpression versus knockdown. And so maybe you can clarify a bit here. They ask, is there any gene that we could target that we can increase its action instead of lowering it? Increasing it seems easier to accomplish than silencing.
So definitely one of the other key modifiers of Huntington's disease called FAN1 is one of these genes where if we were to increase the expression, that would be great in terms of that has a role of protecting and has been shown to protect the somatic repeat expansion. I think for companies, and there's definitely some companies that are working on this strategy for FAN1 raising, but I think for a lot of companies, it's actually easier to lower something or inhibit something than it is raising. It actually is a bit of a difficult process. But for example, like these drugs I talked about, it's pretty easy to design if you've got the right target that you can hit to optimize the compounds to lower a thing.
Yeah, I mean, it seems like it would be easier, but I agree. When you get into the biology of it, it's way easier to lower something than to overexpress.
Yeah.
I hear someone, they say, great presentation, which I very much agree with. And they say, could you tell us more about somatic repeat expansion studies in vitro? If expansion takes decades in human brains, how do you get expansions to happen on a more shorter time frame for research purposes?
That's an interesting question. So yeah, um, so it's true that the repeat expands over many decades in patients. But we do know that the um, the somatic repeat expansion depends a lot on how long the repeat is in the first place. So for in vitro model purposes, if we actually start off with already a really long repeat, for example, the models we use have around 120 repeats, the repeat is actually, when it's that long, expanding pretty fast, especially in certain cells under certain conditions. You can get the repeat to be expanding fast. That's one thing. The other thing is that we can actually do pretty sensitive assays to monitor this. So it's still actually like relatively slow. We're only like when we do our analyses, we're gaining a couple of repeats in our models. But because we've got quite sensitive ways to look at it, we can still track this over uh, weeks, hopefully days, with different models people are making.
And I think Zach's being modest here. He's actually helped develop one of the first cell models that shows very stable repeat expansion. So the RPE cell line, which Zach helped develop. So there's a bit of modesty there that I will put him in the spotlight for. Someone is seeking clarification around PMS1. So hopefully you can kind of dig into this a bit. They're saying, can you talk more about PMS1? Do we all have it, or does it get injected as part of the therapy?
So we all have PMS1. So this mismatch repair pathway is really a fundamental process in our cells. All of our cells are experiencing DNA damage, and so they need to have these ways of repairing the damage. We're constantly being bombarded with damage, and so one of these pathways, like I mentioned, is this mismatch repair pathway. And so PMS1 is a component of this pathway. There's like many different components, like a tree of different components of this. So PMS1 is kind of on one branch of the tree, and we know if we take out that branch, there's still like the rest of the tree, but without that one branch that is PMS1, that's sufficient to stop the repeat expansion, but it doesn't completely destroy necessarily the whole process. And that's also true for um, some of the other targets here.
Thanks, Zach. So this is someone asking, although all cells have mutant Huntington, why do only a fraction undergo somatic CG expansion?
That is a very astute question. It's kind of unknown why certain cells are experiencing this. Um, so we know that the neurons in the brain that are vulnerable, the medium spiny neurons, have the somatic expansion of the repeat, but some of the next door cells, these glial cells, do not. And it doesn't seem to be just as simple as like the expression of some of these key genes. Most of the time they're all being expressed. So it's a really interesting research question. And I think perhaps another like key thing for us to understand this.
Actually, I have a question that builds off of that, and it kind of came to my mind when you were re-showing that branaplam data. Is, we do see these expansions in the MSNs, and once they reach this threshold of 150, we think that's what's targeting toxicity or triggering toxicity. But what's happening with the systemic therapeutics, right? Should we be delivering somatic instability targeting therapeutics systemically if this is only happening in the brain? Do you think we specifically need these in the brain? And would it be detrimental to have it be systemic?
That's a good question. So That's, I think, potentially true. Like, we want to like have this as targeted to the like vulnerable region as possible. Like, um, I think I think there's potential that somatic repeat expansion is playing a role also in other areas of the body that we don't necessarily understand because we're like hyper focused on the brain. But I think it's probably true, especially when we're like tinkering with the DNA repair mechanisms of the cell, trying to get that as specific to the brain. So that's definitely one downside of these drugs. For every therapeutic, you're going to have to really weigh some of the pluses and the minus, and the broad distribution of these drugs like into the brain is actually a pretty significant positive, I think.
Yeah, I think in the fact they can be taken as a pill, right, versus brain
Yeah,
surgery or spinal injection, huge bonus.
yeah.
Okay, so this is someone, they want to know what would be some possible downsides of long-term targeting of PMS1 given its role in DNA repair?
So this is kind of an unknown. So um, there is like with the mismatch repair system, there are certain components of the system that we know are associated with forms of cancer. Um, so like MLH1, some of these other ones, if you have a mutation in these genes, you're higher risk to develop cancer. PMS1 and the other key therapeutic target, MSH3, don't necessarily have these strong associations. And it seems like people can lose this without uh, too much damage. There's also like the idea we can look at it on the genetic level and say how much overall does there seem to be a tolerance to mutations in these genes, and MSH3 and PMS1 seem to be quite tolerant to mutations overall. We also know from some evidence of some very rare people with HD that have inactivating mutations in PMS1 and MSH3. And this is some of the great evidence that, for example, if you have one copy of a loss of function in these, you can really significantly delay onset, and there wasn't a significant other health issues associated there. So it seems to be relatively safe, but it's definitely something that we need to like stay on top of as we develop these therapeutics.
Thanks, Zach. So here's someone asking about off-target effects. They say, are there any concerns of PMS1 lowering causing adverse or off-target effects?
Definitely, like in the drugs that we are talking about here, like branaplam, for example, the reason we were able to figure this out is because it has an enormous number of off targets. Like PMS1 is like one of these off targets for this drug. It's a relatively non-specific drug, but this is like some of the older technology splice modulators people from these companies tell me, and that the new ones that they're developing are better and more specific. Um, so it's part of the whole like drug development process is that when these companies are designing these drugs, you go through, you are looking to see, okay, which one is targeting our target PMS1 the most sensitively while minimizing the potential off targets, which could be super important. Um, Another component of this that we looked at in our study is that you don't only have to think about the off targets in terms of, okay, what are the direct drug targets? We were interested to know if there's genetic variation that is surrounding where these drugs are targeting, does that impact the like splicing, for example, the off-target potential? And we show that yes, there's definitely different patients might have different off-targets is kind of the idea. Um, and so that's kind of like a complex layer on top of this for these companies to think about. Um, for example, that might explain why like some people uh, respond differently to these drugs like branaplam.
Oh, that's interesting. I never considered that. Here's a question that I had, so I'm going to ask it, even though it just came in. They ask, have you ever tested Votoplam to check if it lowers PMS1?
So we haven't worked on that, but I've seen data from other people. For example, at a recent conference, the company Revere Therapeutics they talked about, they showed data that when they treated cells with the Votoplam, it did not target PMS1 at high enough levels that it would have a therapeutic impact. And I think that's also true for Skyhawk therapeutic drug. It doesn't target the PMS1 at It does target it, but not at sufficiently high levels to potentially have a therapeutic impact.
Interesting. Um, so this is a person asking you to kind of opine on the timeline, which I know is always tough, but they ask, do you have an idea about how long symptom progression could be prolonged by targeting both mutant Huntington and PMS1?
So that's definitely unknown. Like I think in an ideal world of therapeutics, you would have this like dual uh, targeting approach where you target both mechanisms of the disease. Um, but how big of an impact? It's really I don't know about that. We can say we can have estimates for Huntington for sorry the like PMS1 and MSH3 targeting, they can have like in the exome sequencing data like a pretty significant impact of like I forget the number, it's like 10 years or something for a heterozygous loss of function MSH3 mutation delaying effect. Um, and people are doing modeling on this now, so um, and that's relatively conservative estimates. But definitely we know from small changes in these modifiers, very small changes, just a couple of percent, you can have like a couple of years over the entire lifetime of the patient.
Thanks, Zach. So here's someone that wants to know what they can do. They say, other than the obvious routes of donating money and sharing information, what are some other ways we can support this important research?
I think there's various ways that you can do it. So I would say it's very important if you're comfortable participating in some of the, like for example, the natural history studies that are studying the HD uh, patient population are super important, like these Enroll HD, some of the biomarker ones I'm forgetting the name of right now. What's that one called, Sarah?
Which one? Shield? Shield HD or
Yeah,
Clarity?
yeah,
HD
participating in that
Clarity?
one. Also participating in, like, hopefully some of these will become clinical trials at a certain point, participating there. Then the secondary thing, like the other thing is like, for example, supporting patient organizations like HDF, HDSA, uh, and yeah,
And I would also add, I know it's kind of like no one wants to think about it, but brain donations have been super duper critical in advancing what we know about somatic instability. The papers that Zach showed from Steve McCarroll's lab, that was all done in human brain tissue. And I think, we can only learn so much from mice and cells in a dish and the animals that we model. And learning what we are from human brains, it's an incredibly it's basically the most generous donation that anyone can make to an HD researcher because it gives us a wealth of information. And I know it's obviously a very deeply personal decision and something that people don't want to think about, but there is the Brain Donor Project that can help set up these things in advance. And it really is the best way that people can advance research, and it has advanced somatic instability light years. I know that.
yeah, definitely.
Um, so this is someone asking, kind of comparing different therapeutics. How do splice modulators compare with other lowering strategies like antisense oligonucleotides and RNAi?
It's probably the really big one is the route of delivery. It's the like I mentioned that the like a pill or like a liquid that you take through the mouth. Um, it's like the really key difference. Some of the ASOs are like delivered through injections into the spine, um, and some of these other therapeutics need to be like delivered directly into the brain. So these therapeutics are a much lower barrier to entry, and a much less daunting thing, I think, for people. If you just have to take a pill, it's like much much easier to imagine that. Whereas some of these other ones, it's like a much bigger commitment. And also sometimes also like, for example, if you do a gene therapy one, it's like a irreversible situation. You can't go backwards. Whereas like these ones, for example, the patients that were in the branaplam clinical trial could just stop taking the drug, and the symptoms were that they experienced from the side effects of that drug were reversed from stopping taking the drug.
Thanks, Zach. So this person is asking about the germline. So will inhibiting MMR proteins affect expansion in the germline?
That's an interesting question. So I mean, somatic repeat expansion, I think, is a shared process in the germline and the brain. Likely, they have the same modifiers that modify the process. So I think it's pretty likely if you have a drug that is spread around the body that is also um, going to have the same effect in the germline. Um, One issue though, I think, is like the um, yeah, I think that's the case.
But I think it's probably important to clarify that it isn't necessarily, it wouldn't change like passing it down to your children, right?
Yeah,
Like there
it's
would
still
still
going to
be
be
the
an
50%. Yeah,
exactly. It's still a 50-50 chance. The repeat isn't going down. It's just halted where it is. Yeah.
I think that's an important part. Thanks. So this person asks, why can you only see somatic expansion in the brain, and why is blood CG repeat results used for eligibility for trials or looking at disease burden? So I
Wait,
think
sorry.
what they're getting at, yeah. So I think the difference between these expansions that we see in the brain, but we're using CAG size from blood samples to get into
Right,
trials.
right. It's just a fundamental issue. The cells that we really want to know the repeat length of is like neurons in the brain, and we can't easily take a sample of that. So like we're going to take a sample of the blood. We know from studies that we can actually detect the somatic repeat expansion in the blood. It's like very, very minor relative to what goes on in the brain, but we can detect it. And when you do studies, for example, where the GMHD consortium led the study where you look at the somatic repeat expansion in the blood, you can identify the same genetic modifiers that it was somewhat overlapping list that is associated with the age at HD onset. So the blood can be a biomarker for the somatic repeat expansion, I think, that's happening in
So
the neurons.
thanks. So here's someone who asks, does late onset HD run in families? Could it be because of these genetic differences?
I think, yeah, I think that's likely an explanation. Um, um, Having a shared genetic background um, in this polygenic situation, so meaning that like you might pass down, so the HD gene might be passed down, but then protective genetic modifiers might also be passed down that delay, for example, the rate of somatic repeat expansion. So I think there's probably an explanation for that, and that's kind of what the GWAS is saying, isn't it?
That's interesting. So I think this might be a good one to end on because I think you've kind of summed all this up, but this asks it in a very pointed question. They say, thanks for the great talk. Why do MMR proteins only have an effect on somatic repeat expansion in certain cells, like neurons, but not like neurons and MSNs, but not other neurons? In general, why is repeat expansion limited to certain types of cells?
So, like I was saying before, this is like a really interesting question. It's perhaps like a balance of all of like it's an intricate system of players in the somatic repeat expansion process. Perhaps there's like a certain balance of these, and if you have just the right balance, then you are resulting in the somatic repeat expansion. But it, like I said before, it doesn't necessarily look like it's just the expression of the the modifiers that we know of. It doesn't seem to be just as simple as that. There seems to be another component that we don't understand necessarily.
Okay, well, thank you so much, Zach. I think the award goes to you for the absolute longest Q&A that we've ever had on one of our webinars. So I appreciate you answering all these questions. And there's actually some we still didn't get to. So people are very interested in your work, and I appreciate you sharing it. I'm going to end by thanking our sponsors, Ionis Pharmaceuticals and Unicure, as well as everyone who donates to the HDF to support the research that we fund. And of course, everyone for joining us. Thank you so much for tuning in. Last but certainly not least, I want to thank Zach. Thank you so much for taking the time to share your work.
Thanks, Sarah.
Our next Our next webinar is Tuesday, May 20th. During HD Awareness Month, we are going to be joined by Dr. James Gusella, who was the winner of HDF's 2024 Leslie Gary Prize for Innovation in Science. And that is just one piece of the exciting programming that we have planned for HD Awareness Month. HD Buzz will also be taking over HDF's Facebook page on May 7th. So make sure that you follow HDF on the socials so that you can get live access to HD Buzz and ask them any research or science question that you have. Registration for our May webinar is now open, so head over to the HDF website right now so you can get it on your calendar. Thank you so much for joining us, and I will see you next time during HD Awareness Month.